Hello, my name is Ed Chapman, and this is part 2 of our tour of the cell. And we're going to start with the endomembrane system, which is a set of organelles that are connected to each other and interact with each other throughout the cell or throughout the cytoplasm of the cell. And there are three main components of the endomembrane system that you probably have already heard about in freshman biology. There's the endoplasmic reticulum, the Golgi apparatus, and the lysosomes. Now, the endoplasmic reticulum has two forms, rough and smooth, and we're going to take each one by one and talk about its structure and its function. The rough endoplasmic reticulum is called rough because it is studded with ribosomes, which are attached to its outer surface. And the ribosomes are where proteins are being synthesized from amino acids. And once the proteins are assembled into chains called polypeptides, these chains of polypeptides then travel into the inner chamber of the rough ER where they get folded into proteins. The other part of the ER is the smooth ER. And the smooth ER looks smooth because it doesn't have ribosomes attached. And the smooth ER is also responsible for synthesis inside the cell, but it's synthesizing things like lipids and lipid hormones like estrogen. It also metabolizes carbohydrates, which usually means it helps in the process of digesting carbohydrates. And it also detoxifies drugs and poisons. So, for example, your liver does a lot of detoxifying of materials in your bloodstream. Therefore, your liver cells structurally should contain or do contain lots of smooth ER. The Golgi apparatus looks a lot like smooth endoplasmic reticulum, except it's more like a stack of pancakes or a stack of flattened vesicles stacked very closely to each other and kind of folded around each other. It's where materials are going to be sorted and finished, packaged for export out of the cell. And the Golgi apparatus has two sides, a side that's taking in vesicles and a side that's releasing vesicles. The cis face of the Golgi apparatus is where material containing raw materials enters the Golgi apparatus, kind of like bubbles coming into a lava lamp. The trans face is where materials are exported out of the Golgi apparatus, and they contain finished materials that are going to be fusing with the cell membrane usually and leaving the cell. The cis face is illustrated here, and here is the trans face. Okay. Lysosomes are specialized vesicles for digestion. They contain enzymes that are hydrolytic, which means they can hydrolyze polymers into monomers. And lysosomes are compartments where this digestion can take place without damaging or risking damage to the rest of the cell or the cell's organelles. Lysosomes are capable of digesting proteins, carbohydrates, lipids, just about anything. And their environment is acidic. The acidity is one of the ways that the digestive enzymes are regulated by the pH inside the lysosomes. Now, lysosomes can do two basic kinds of digestion. They can digest food vacuoles, which means the cell absorbs something, surrounds it with membrane from the cell membrane. This food vacuole then fuses with a lysosome so that the lysosome's digestive enzymes can mix with the food that's been surrounded by the cell membrane, and together this food vacuole is where digestion happens. Also, the lysosome can do something called autophagy, which means it can absorb broken or worn out cell parts and break them down and recycle their components. In this picture, you can see a mitochondrion that's been sort of quote-unquote swallowed by a lysosome, and it's in the process of being digested or its parts recycled. Lysosomes are formed either from the rough ER by way of the Golgi or from just the rough ER, depending on what their function is. Here you can see a bacterial cell being endocytosed or taking in by the cell membrane, and then this food vacuole which forms around the bacterium is then fusing with a lysosome for digestion. You can also see a different type of lysosome fusing with a mitochondrion to digest it. But I think you get the idea. Lysosomes are like little stomachs inside the cell or little garbage disposers inside the cell. They take things apart using digestive enzymes. Mitochondria and chloroplasts together are the two energy transformers of eukaryote cells. All eukaryote cells that are aerobic have mitochondria, and some of them, which we call plant cells, have chloroplasts. They are for example, the mitochondria are compartments for cellular respiration. And this means that they're responsible for a catabolic metabolic pathway. They take apart large molecules and release energy in the process. Mitochondria are ATP generators, and they're extracting energy from carbohydrates like glucose, plus other things like fats, in a process that requires oxygen. Mitochondrial structure is made up of a series of membranes. We have an outer membrane and an inner membrane that has been folded many, many times to produce lots and lots of surface area. Between the outer membrane and the inner membrane is a space called, logically, the intermembrane space, and deep inside the inner membrane is a region called the matrix. So if we arrange these from outside to inside, imagine you're something traveling into the mitochondria, you can see the order in which you're going to encounter each of these membranes and these areas. Chloroplasts are the other energy transformers, and of course they're transforming light energy into chemical energy, and they're doing a process called photosynthesis, which is anabolic in nature. They are harvesting sunlight or energy from the sun, and they're transferring some of that energy to ATP molecules and then using the ATP molecules to build carbohydrates like glucose. Now, chloroplasts are also built as a series of membranes within membranes. Chloroplasts have an outer membrane, then an inner membrane, and then inside the inner membrane are stacks of thylakoids called grana. And grana are where you see the dark green chlorophyll pigment. And between the grana and the inner membrane is a space called the stroma, kind of like a liquid filling, and then inside the thylakoids themselves is the thylakoid space. We'll talk more about this when we study photosynthesis in detail. Again, outside to inside, these parts are placed in order for you. Peroxisomes are the last organelle we're going to talk about in this slideshow, and they're a strange type of vesicle. As you can see in this picture, they contain sometimes these crystalline structures of enzymes, and their job is they're a single membrane compartment, and their function is going to be to take hydrogens off of other molecules. Okay, for example, they rip hydrogens off of fatty acids, or they rip hydrogens off of toxin molecules to change them or digest them. These hydrogens that are removed are then added to oxygen to make a kind of a dangerous molecule called hydrogen peroxide. And peroxide is something that cells don't need a lot of floating around. So another job of the peroxisome is to then break the peroxide down into water, which of course is harmless. That's the end of part 2. We'll pick up with part 3 in the next slideshow.